So hello everyone. In today's video, we are going to talk on interview questions asked by the interviewer for QA/QC microbiologist post. So before starting the actual part, one request is there to you people that kindly subscribe to my channel and like and share my videos. So let's talk about the interview. So whenever you are going to face an interview, make sure you have prepared well for that. In interview, questions are asked related to your work that you are supposed to do after selection in that company or in that field. Okay, so they won't ask details of work, but they will definitely ask you basics of that work or post or job. Okay, so for QA/QC microbiologist post, interviewers will ask questions regarding the basic concept of microbiology, specifically pharmaceutical microbiology, microbial testing, microbiological skills and practices, and they will definitely test your knowledge regarding analytical techniques, lab practices, and regulatory practices in pharmaceutical industries. So today in this video, we'll discuss few questions that may be asked in the interview by the interviewer. So I'll tell you questions and their answers one by one. So the first question that may be asked by the interviewer is, what is sterilization? So this is very basic and important question and concept in microbiology. So what is sterilization? Sterilization, it is a process which destroys all the forms of microbial life, including fungus, viruses, and viable cells of microorganisms, along with their spores, by using chemical as well as physical methods So what you do in sterilization, you kill all forms of microorganism, including vegetative forms, viable cells, and spores. Okay, so this is in sterilization. So let's talk about the next question, which might be asked in the interview. So second question is, what is disinfection? So disinfection, it is the process of removing microorganism, including potentially pathogenic ones, okay, from the surfaces of inanimate objects. Inanimate objects means non-living objects and surfaces. Okay, so disinfection destroys or irreversibly inactivates most pathogens. Okay, that includes bacteria, viruses, and fungi. And this destruction, it occurs on non-living surfaces, that is inanimate objects. Okay, so in disinfection, what happens generally? In disinfection, we reduces the number of microorganisms present to a level at which they do not present a risk. So this is the second question, possible question, what is disinfection? Now let's talk about the third question, which might be asked by the interviewer. It is the, what is the difference between sterilization and disinfection? Actually, you should know the difference between sterilization and disinfection, because what happens in sterilization and in disinfection, in both these processes, you kill the microorganisms. Okay, you use physical or chemical methods to kill the microorganisms. But what is the difference? So the third question is, what's the difference between sterilization and disinfection? So in sterilization, you completely destroy all forms of microorganism, including spores. Okay, but in disinfection, you only destroys or inactivates harmful microorganism, that is pathogenic microorganism, to the safe levels. Okay, but in disinfection, this process, it does not eliminate spores. Okay, so this is the difference between sterilization and disinfection. In sterilization, there is 100% killing of all forms of microorganism. That includes vegetative forms and it also includes spores. Okay, and in disinfection, you only kill the pathogenic microorganism. There is you say that there is not 100% killing of microorganism occurs in this process. Okay, so next question which may be asked by the interviewer is, give some examples of gram-positive and gram-negative bacteria. So examples of gram-negative bacteria, you should remember the examples: E. coli, Klebsiella pneumonia, causative agent of pneumonia. Okay, so Klebsiella pneumoniae, this is the causative organism. Then Pseudomonas aeruginosa, Salmonella enterica, Vibrio cholera, Neisseria meningitis, and Salmonella typhi. And gram-positive bacteria examples are Staphylococcus aureus, Streptococcus pyogenes, Bacillus anthracis, Clostridium tetani, Lactobacillus lactis, Corine bacterium, diphtheria, and Mycobacterium tuberculosis. So these are some example of gram-positive and gram-negative bacteria. So the next potential questions from the interviewer could be, tell us about yourself and your experience in microbiology. So in this case, if you are a fresher, you should say in this way, like, I'm a recent graduate with a degree in microbiology where I gain hands-on experience in laboratory techniques such as microbial culture, staining, and biochemical testing. During my studies, I worked on various projects involving microbiological analysis and quality control methods. Okay, I'm eager to apply my theoretical knowledge in a practical setting and contribute to the QA/QC processes, ensuring high standards of safety and quality. I am a fast learner, detail-oriented, and passionate about pursuing a career in microbiology. Okay, so this is how you should answer if you are a fresher. If you are an experienced one, then you should say, like, I have a strong background in microbiology with experience in both research and quality control settings. I have worked extensively with microbial testing, sterility assurance, and environmental monitoring. My experience includes conducting routine microbiology tests, analyzing results, and ensuring compliance with regulatory standards. I'm skilled in using laboratory equipment and techniques such as PCR, microbial identification, and endotoxic testing. And I am committed to maintaining the highest standards of quality and accuracy in my work. So, this is how you should answer the question. If you are asked, tell us about yourself and your experience in the microbiology. Okay, so the next question is, what is QC? Okay, QC, it stands for quality control. So what is QC? It is a systematic process aimed at ensuring that pharmaceutical products are consistently produced and controlled according to quality standards. This is very important, quality standards in pharmaceutical industries. Okay, so this QC, it encompasses planned and documented activities to guarantee the quality, the safety, and the efficacy of products throughout their life cycle. Okay, so this is the QC. Now, this quality control, it is the inspection phase or sub-part of quality assurance. Remember, this is a sub-part of quality assurance. Quality control, it comes under the quality assurance. Okay, so this quality control, it's a series of test procedures used to verify that a product is safe and effective after mass production. So these tests and procedure, they are carried out in the laboratory in that pharmaceutical industry after that product it has been produced. Okay, and after those tests and procedures, it is tested that the produced product, it is whether it is safe or if and effective for the consumption. Okay, so QC, it is all about the inspection, measurement, and testing of your products and processes to detect and correct deviation from desired quality levels. Okay, so this is about the QC. So the next possible question, the interviewer could ask is, what is QA? Now you know what is QC, you should answer what is QA. So QA is, it stands for quality assurance. So quality assurance involves the broad bundle of systems and processes that interact to ensure quality products. Quality control is the targeted nuts and bolts checkup of the products as they are being produced to ensure that QA plans are corresponding to the reality of your manufacturing output. Okay, so in short, QA involves processes and procedures that promote consistent quality throughout the product or service life cycle, and QA aims to prevent. So the next question, possible question, is what is the role of microbiologist in QA and QC? Okay. So the role of microbiologist in QA and QC is to ensure the microbiological quality and safety of products by conducting tests. Okay, this is important, microbiological quality and safety of the products. Okay, then to detect and identify microorganisms, whether they are present in the environment or whether they are present in the manufactured product or not. Okay, so this is the second role. Then third is performing sterility and microbial limit test, then monitoring environmental conditions. Okay, so these microbiologists in QC and QA, they help ensure compliance with regulatory standards, troubleshoot issues related to contamination, and maintain the integrity of processes through proper testing and documentation. Okay, so this was the sixth question. What is the role of microbiologist in QA and QC? Now, next question is, how do you validate sterilization processes? Okay, so in pharmaceutical industry or in microbiology, sterilization processes are validated by performing tests to ensure they effectively eliminate all microorganisms. So generally biological indicators, physical indicators, and chemical indicators, they are being used in the pharmaceutical or microbiological lab to validate the sterilization processes. So biological indicators. So additionally, validation involves ensuring equipment, calibration, and documenting results for compliance with regulatory standards. Okay. So next question, which might be asked in the interview, it is how quality is controlled in pharmaceutical industry. So in pharmaceutical industry, quality is controlled through a combination of quality assurance and quality control. Okay, so these two practices, they are responsible for controlling the quality in the pharmaceutical industry. So quality assurance and quality control. Now, quality assurance ensures that processes, procedures, and systems are in place to meet regulatory standards, while QC involves testing raw materials, in-process materials, and finished products to ensure they meet specified quality criteria. Now, key aspects include in this system testing, which involves routine checks for purity, potency, and stability. Then second is process control, which involves monitoring and controlling production to maintain consistency. And third, documentation. Okay, this is very important in pharmaceutical industry, documentation, keeping detailed records of production and testing to ensure traceability. Okay. And last is compliance, adherence to GMP, FDA, and other regulatory standards. Okay, so together these methods, they ensure that products are safe, effective, and of the highest quality. Okay, so next question is, what is pharmacopoeia in pharmaceutical industry? So pharmacopoeia is an official publication that sets standards for the quality, purity, strength, and consistency of medicines, active ingredients, and excipients. Okay, remember this is very important. Pharmacopoeia, it is like kind of, it is a book, okay, which have rules and regulation for the you can say quality, purity, strengths, or various kinds of procedures. Okay, so it provides detailed specifications for drug substances, formulations, and methods of testing to ensure that pharmaceutical products meet required safety and efficacy. standards. Okay, it is a book describing drug, chemicals, and medicinal preparations, especially one issued by an officially recognized authority and serving as a standard. So this pharmacopoeia or kind of book, it is issued by an official authority and it served as a standard. Okay, so example include United States Pharmacopoeia, European Pharmacopoeia, and Indian Pharmacopoeia. Okay, so this is the question, possible question which might be asked in the interview. Okay, it was regarding the pharmacopoeia. Now the next question, it is about what is GMP. It is very important in pharmaceutical industry. GMP, good manufacturing practices. Okay, now these practices or GMP, it is a set of guidelines and regulations that ensure products are consistently produced and controlled according to quality standards. Okay, now GMP covers all aspects of production, including personnel, equipment, facilities, and raw materials to ensure the safety, efficacy, and quality of products, especially in the pharmaceutical, food, and medical device industries. Okay, so this GMP, you can say these are the set of group of guidelines and regulations, okay, which ensures the products which are being formed or manufactured in the pharmaceutical industries, they are produced under the by using the quality standards. Okay, so GMP covers all the aspects of production, which includes staff members like personnel, equipment, facilities, and raw materials. Okay, now next question, probable question is what is GLP? So GLP stands for good laboratory practices. Okay, so what is GLP? It is a set of principles and guidelines ensuring the reliability, consistency, and integrity of non-clinical laboratory studies. So in this case, GLP, this is for the laboratory practice, good laboratory practices. Okay, so GLP is a set of principles that are designed to guarantee the quality of non-clinical laboratory studies, ranging from in vitro studies to preclinical animal experiments. So these are the some questions which could be asked by the interviewer in the interview. So please go through these questions and prepare well for the interview. Thank you very much.